In this lecture, I just want to discuss some of the methods and techniques that we can use to explore and examine DNA and chromosomes as we move into and work into RNA processing and some of those other techniques that you're going to describe in your presentations. So the backstory of this figure is that originally scientists believed that protein was actually their hereditary source used to transfer information from one cell to another. And the reasoning, the rationale was actually not a bad one. It was, they were looking at the amino acids, right? There are 20 different essential amino acids, and there are only 5 types of DNA or RNA, right? So they were kind of thinking, well, we have all of this complexity, all of these different types of things that, DNA can make up, or that protein can be made up of and can change, where DNA just is so limited, so it doesn't seem like that would be the hereditary source. So we're going to talk about that and understand one of the things that they could do to test that theory. So what they did is they took a bacteriophage. So this is a virus that basically infects bacteria. So here's an E. coli bacteria. Here's the virus, and the genetic material is going to be transferred from the virus to the bacteria, and then that will be, moved on into another viral particle. It will get synthesized in this E. coli cell. So this bacteriophage was used to show definitively that DNA, not protein, was used as the source. Now we know that 32 phosphorus, so this is a radioisotope of phosphorus. We talked about phosphorus at the beginning of the year. If we had DNA labeled with 32P or P32, it would get incorporated into only the DNA, because we know that in the phosphodiester bond, that's going to be found in the nucleic acid. And if we have sulfur 35 is another radioisotope of sulfur, so it's a heavy labeled sulfur group that is going to be found in cysteine, but it's not going to be found in DNA. So these two, having them labeled with heavy phosphorus or heavy sulfur, that would allow us to figure out where the genetic material is coming from. So what I want you to do is take a second, I want you to pause this, and I want you to draw a DNA fragment and circle the location that the 32P would be incorporated, and then draw the amino acids that would be labeled by S35, and then is there phosphorus in protein and is there sulfur in DNA? Those are kind of the questions I want you to make sure that you understand before you move to the next slide. So here's the experiment. We have both phages with P32 and S35. We incubate it with E. coli. The viruses infect and integrate their either their radiolabeled phosphorus or sulfur, and then they're chopped up and everything's kind of sheared off, and the E. coli is spun down. The viruses these are a centrifuge, and the virus heads kind of get in the supernatant, and the cells are pelleted. And then what's your prediction? What will be in the supernatant? Where the free viruses will be? What's going to be in the pellet? What is that going to be? What's your prediction? And before you move on, just kind of try to think through that. What's going to be, where is the phosphorus or the sulfur going to be located? Okay, so what you should have found out was that the infected bacteria contain the phosphorus but not the sulfur. So that's kind of what we were expecting. Earlier we were describing, because the phosphorus is found in the DNA, the sulfur is found in the protein, that would have been in the supernatant with the labeled virus perhaps. So we know that the chromosomes are stored in the nucleus of the cells, and that we have all of this complex structure. So we have the base pairs that we've looked at, the structure of those, and the phosphodiester bond, and the sugar backbone, and the bases that attach to each other. But the bigger macromolecule that's formed, we have the double helixed chain that's going to be attached, and these are surrounded by histones that are proteins that we'll discuss in a little bit, and they're wrapped around these proteins that allow us to have this higher ordered structure that create this larger chromosome and organizes the different chromosomes and the chromatids. So here we have labeled DNA, and I just want to give you a chance to look at these figures here. So this is under a microscope, we would be able to image these different chromosomes. So every chromosome in a human is different and unique to the other ones. So this is kind of how it would be normally organized, and then using computational methods, they can organize and sort all of these different colors of these chromatids. And I want you to guess, how do you think this assay is done? What kind of technique might you use to do this? And I just want you to write down what you think. It doesn't have to be right, but I just want you to consider that before you move on to the next part. Okay, did you write it down? Well, here it is. What you actually do here is you have DNA that is hybridized with fluorescent probes, and there are different probes of different wavelengths. And this is called karyotyping. So it's done a lot in cell research, also during pregnancy. During higher risk pregnancies, they might take some sample tissue or skin samples or amniotic fluid from the growing fetus, and you can make sure that it can be done to make sure that's not harming the child or anything, and to make sure there are no duplications or changes or abnormalities in the overall karyotype. But what happens is you have these DNA molecules with these fluorescent probes that hybridize onto specific components or specific chains in here, and then they hybridize, and then you can identify for example, in humans, the 23 different chromosomes that we have. Here's an example where it's not fluorescently labeled, but there's it's the same idea. It's DNA probes with a different type of modified component that's not necessarily fluorescent, but it's looking at, you're able to still distinguish these different types from each other. Here you can see there's a trisomy, so it's a whole entire chromatids duplicated. So this is trisomy 21. That's typically what happens with children with Down syndrome. It can happen to different ones. So if you have trisomy in 13, that's Patau syndrome. In 18, that would be Edwards syndrome. So there are a lot of different types of syndromes and things that can happen when you have extra chromatids, and that can mess around with the amount of the mRNA that's transcribed and the some of the other functions of a cell. So it's not always a bad thing. This is how the evolution occurs as well. So abnormalities during chromosomal separation can oftentimes vary over long periods of time. They can actually separate types of species. So here's the Chinese muntjac and the Indian muntjac. They have similar DNA. If you actually look at the similarity of their overall genome, it's extremely similar, 99+%. But you can see that they're broken down into smaller bits in the Chinese muntjac, and here the Indian muntjac, there is some type of abnormality that happened, and they're very similar types of animals. They have similar characteristics. It's just spread out differently over different chromosomes. So this Indian muntjac has to kind of just manage these three sex chromosomes a little differently than this Chinese muntjac. So they wouldn't be able to mate because of these large differences in how their DNA is organized. Again, that chromosomal change for species separation. Here is a female human and a male bonobo monkey. You can see that they have a few more chromosomes. So they have 24, we have 23. You can kind of see how we have some changes here that are occurring, but largely similar, largely the same, but there's some different movement in where those chromosomes come together. Of course, for each of these, right, we get one of these comes from our mother, one of them comes from our father. So in this case, for the female, one of these X's came from the dad, one of them came from the mom. Here, the Y had to come from the dad, and the X came from the mother. So in females, we don't know necessarily which unless we know what the father's genome looks like, or which one would be which. And there's also, we'll talk about homologous recombination, but this is kind of the setup for we are getting one of these from mom, one of these from dad. This is typically where abnormalities happen during that homologous recombination when we have cell division happening. And here's an example of an abnormality caused by chromosomal swapping. We have this addition where we had, actually that last slide, we can kind of see some of these 4 and 17. If these things are flopping from one chromosome to another, these can cause some problems. Here's an example, for example, where we're moving an addition of chromosome 4. This is this red section here, which is that section down here, onto 12. So we're elongating this one chromosome 12, and it's causing problems here. So this actually causes ataxia and motor movement problems for this recipient. So genetic information is pretty tightly packed onto the strands of the chromosome. And here we have, in yeast, we have about over 6,000 genes on 16 chromosomes. You can see that most of the genes are found on one side, but you do see some sprinkled in here found on the other side of the chromosome here. Genes generally encode for proteins, so they are generally encoding for a messenger RNA that can then be synthesized into proteins. There are also non-coding RNAs, so that means that they can still be transcribed, but they're not necessarily translated then. So these include RNAs that have different functions, such as small interfering RNAs, siRNAs, microRNAs, miRNAs, small nucleolar RNAs, that's SNO-RNA. We also, that are involved in splicing and some of the other processes. Some of them are involved in, they're part of the ribosomal RNA groups that are a part of that, or transfer RNA. So there are a lot of different types of RNAs that also need to be transcribed from our DNA genome. There are different types of areas on the DNA, and these are sequence areas. So these different regions are recognized by the sequence that are encoded in the DNA here. So we have telomeric binding sites, replication origins, where we have DNA sequences that define where we're going to, during mitosis, for example, where we're going to duplicate the DNA. Centromere sites, where the mitotic spindle will bind to and recognize. So these different sites that are in blue and yellow and red, these are actually sequences in the DNA that are recognized by different proteins and different things that are using this and helping to duplicate it, or during cell division. They're necessary, important, and sufficient for doing these different things. Here's an example of a chromosome during replication. So this would be splitting up, right? They're kind of pairing up, and then this is being replicated on the other side here. So that has to do this while the cells are being divided. All right, quick question. What kind of microscope could take this image? So do you know the answer to that? I'll let you write that down and make sure you can figure that out. So that last image is kind of the typical chromosome figure, or, that you'd think you would kind of see. But normally they're not necessarily in that orientation. That's more happening during cell division. But even when they're not actively replicating, they do remain compact. They're not like spaghetti in a pot, so it's not like they're just kind of all over the place. So they can actually be attached to the nuclear cell wall, and they're typically organized within the nucleus and have that organization is important for the function to organize different types of components to allow them to be actively translated transcribed, and then that is going to allow for things to be compartmentalized and compacted more, where it's not being maybe used as much, depending on the type of cell that is activated. Here is an image of looking at the nucleolus, where these ribosomal RNAs cluster. So these are going to be kind of forming here. And this is the heterochromatin, so the differently packed, these are different densities basically of these different DNA molecules. So ribosomal RNAs will be on multiple genes, but they usually cluster together in the middle here. Another quick question, what kind of microscope could take this image? So it's different, right? That other one was more three-dimensional and this one's kind of more flat. What two different microscopes can take images like that? One major class of protein that binds to DNA are called histones. There are other types of non-histone chromosomal proteins that also bind to DNA. We'll talk about both of these. All right, what kind of microscopy can image these things? We're looking at DNA and nucleosomes. So we have DNA that's kind of spun around a core of proteins that are histones. So you can see this kind of chain here, these beads of, these are partially treated, unfolded here, and you can see the nucleosome beads. So these proteins around the histones. And here's more compact, where these are kind of spun around each other a little more densely. So this is kind of the cartoon image of what we're seeing down here. We have these linker DNA, so this double-stranded DNA are spun around these histones, and these are kind of, you can see that when we digest these, we get these longer pieces here. You can figure out about how many nucleotide pairs of DNA would be included in spinning around by digesting the linker DNA and then looking at how much is left here. So it's typically about 200 nucleotide pairs. You can kind of see here when we're looking gentle digestion, we can use that to kind of figure that out. So we cut basically between this phosphodiester bond in order to do that. The reason that all of this doesn't digest, if we're using a gentle, short, kind of quick digestion, is that these proteins actually protect the DNA that's wrapped around it from being digested. So that's part of the reason to have these, is they're protective of the DNA, and they help to kind of keep it from getting digested or broken down as easily. To study histones, you can unwrap this with high salt, so that would break down some of the intermolecular forces, some of the interactions that the histone and the nucleotide, the double helix, are making together. And then you can break down those dimers even further apart from each other. So these are, you can see, held together again by quaternary structures. So these are multimers of different proteins together. So then you can run mass spectrometry or something to identify what type of proteins those are. But there are 4 different types: H2A, H2B, H3, and H4. So 4 different types of proteins that make these histone proteins. All right, so this is a fun little quiz question. Why do you think it's important for histones to have a lot of positively charged amino acids? So what could be the value of the positively charged amino acids? So histidine, sorry, these are a little grainy, but histidine, lysine, and arginine, right? This is the backbone, the NCC, those are backbone down here that are going to be part of the protein. So even those are positive and negative, they're going to be uncharged if they're a peptide bond. But the positive charge is here. So why is that important that they're positively charged? What is the value of that, do you think? So I'd like you to answer that question before you move on. And let me know if you're not sure what that question is or what the answer is when we meet in person. So an interesting way that the histones can work is that they can be modified. So they can be, it's called post-translational modification. So it's a modification on the protein that can change its function. And you can see here in gray, this is the DNA helix in gray. And then in green is the H3, in blue is H4, and then red and orange are the H2A and H2B. You can see that these fit together, and the H3, that histone tail sticks out, and that can be modified, and it'll change how these things are able to be packed together. Here's an organized diagram of some of the modifications that can happen, and typically we're looking at serine, arginine, and lysines are found to be modified. So arginine, lysine, serine, you can see that those those numbers, or the single letter amino acid, and then the number of that chain length. So we're at 2, 4, 9, 28, 36. So that's the number, that's the length of the protein. This is where it's happening, typically on the end, on the tail, not necessarily the core of the protein. And here we have typically acetylation is happening in purple, methylation, even phosphorylation in gold here. So these are some of the common modifications that can happen. Here are some what happens. So there are a lot of different places these can be modified, and it's not always known why or why that's getting modified or what the purpose is, but there are some that have some meaning that we know about. So on lysine 9, if that's methylated, that typically creates the formation of heterochromatin, which is more compact, tightly pulled together chromatin, and then that helps to silence the genes. So when things are kind of wrapped up really tightly, it's hard for promoters or different things to get in there to activate those genes. When we acetylate that same lysine and then methylate a different lysine on that same chain, that can open it up, and it can basically loosen up that heterochromatin, and then that helps to promote gene expression. Also in serine 10, phosphorylation, lysine 14, acetylation, that can also promote gene expression. So different things that we're learning about this histone modification and the influence of gene expression. Just looking at this change, what's happening here with the methylation, we can see that the lysine gets methylated and it loses that positive charge. So these might pack together more with itself and less with the DNA. So it'll change the interaction with itself and with the DNA that's it's associating with. Here's an example of the lysine to acetylation. So we changed some of the different, how that structure of that looks. And histone acetyltransferases, histone deacetyltransferases, those are the enzymes that are involved in that process. So they can be acetylated, methylated, phosphorylated. They can also be sumoylated and ubiquitinated. So a lot of different types of modifications, and there's some work being done to really understand how these different signals and the changes influence gene expression. And here are just some known modifications on human histones, and understanding these patterns and understanding how these things work and why they're happening can help us understand why we have different gene expression profiles. This can change, how you look or what your hair looks like, or a lot of different things, right? They can change a lot of the expression of protein, and that's also ultimately going to change how you behave and who you are. So these are really interesting things that can help us understand more about cells and more about ourselves. Likely a lot of those different modifications of the histones themselves, those tails, are actually recruiting other proteins. So those modifications can recruit other chromosome modifying proteins. For example, ATP-dependent chromatin remodeling complex, which is right here in green. So those different modifications on this histone can help to recruit or, not recruit these kind of things. So this is what's going to be, if we're looking at heterochromatin or condensed chromatin, if we have a remodeling complex that comes in that can be associated with the chromatin, and it can help kind of unloop this and decondense the chromatin to allow it to be more easily expressed. So if it's really tightly wrapped, it's hard to get in here. If it's loose, it's unpacked, then you can get in here and express that. So it's kind of like putting Christmas ornaments away, right? If you want to take them down off the tree, you wrap them up, you put them away, and then when you want to use them, you unwrap them and they're more available. Here's how heterochromatin can spread. So you have this heterochromatin that's been modified. You have euchromatin, which is opened up a little bit, and then the sequence that kind of keeps it from getting compressed further. So what happens is these things are modified so that, for example, we talked about the ubiquitination or acetylation. That'll modify the tail and that'll help recruit different types of proteins to come in and then actually compress and condense more of this euchromatin into heterochromatin. If it hits a barrier, it hits a sequence that maybe has other proteins that are going to stop that, then that will keep it from kind of moving into the next section of DNA. Here's how it's all kind of packed together from little to big. We have double-stranded helix of DNA wrapped around histone proteins. Those proteins are lightly packed into chromatin fibers in the nucleosome. Those fibers are then further packed into these loops and bundles in the chromatin, and then that's just one section of the chromosome or the chromatid half, right? So these are going to be kind of how this all fits together. So the net result is a ton of DNA packed into a small little area that can be managed, divided, and split up when it needs to, and then it can be opened up and expressed also when it needs to be done as well. Here is a scanning electron micrograph of a DNA chromatid 1, chromatid 2, two different sections of a piece of a chromosome. So here's another example of changing the DNA structure and how it can really change gene expression. Typically, we know men have two X chromosomes, men have an X and a Y. And we also talked about trisomy, right? So sometimes the overexpression of certain types of information can be detrimental to the system. Women get around that issue is that they'll deactivate one of their chromosomes. So they're deactivated so they don't get a double dose of genes, and it's packed into heterochromatin and it's stored. And then this is the way, while other chromosomes can be used and expressed for genes as needed. So typically this is an interesting thing that happens in cells where it'll select one versus the other, and then that gets turned into the heterochromatin, which is kind of shut down. And so you might have different gene expression, and this can happen in tissues or cells. If you look at different types of animals like cats or dogs, you'll notice that they have some patterns that may be different, that might be due to different regions of heterochromatin. So different colors are going to be expressed in different areas of their body based on how these are expressed. Calico cats, right, will shut down some genes versus the other in certain parts of their hair tissue, so they'll have different colors throughout their body, typically because of this type of situation where we have heterochromatin of certain genes are being shut down in certain areas of the body versus others. It's a pretty interesting phenomenon.